It gives me great pleasure to introduce to you one of the most famous vascular surgeons in the world. Think of that. That's a bold statement. Dr. Ron Fairman is here with us today to talk about clinical trials for abdominal aortic aneurysms, a 20-year experience. And I'll tell you a little bit about Ron. Ron grew up in the Philadelphia area. He went to Muhlenberg College. Does anyone know where Muhlenberg is? Allentown, Pennsylvania. And he then went to Jefferson Medical College. The reason that's important to me personally is because I went to college at a very small, unknown place a little bit to the west of Allentown in Reading, Pennsylvania, called Albright College. And then I went to Jefferson Medical School. So if you triangulate those three cities, you find the King of Prussia, Maul. And where is that? That's where Villanova is. So Villanova just won the national championship, and I congratulated Dr. Fairman when he got off the plane for that deed. He has had an incredible career. After attending Jefferson Medical College, he went to the University of Pennsylvania for his residency and his fellowship in 1984. It was one of the first accredited fellowships in the nation. He then went into private practice for 12 years at Jeanes Hospital. He was a clinical associate at the University of Pennsylvania. And he decided when the endovascular revolution was occurring that he needed to be in academia. So after 12 years of being in private practice, he went back into academia and had an incredible career. Still does have an incredible career. He is the chief of the Division of Vascular Surgery at the University of Pennsylvania, and he has been there ever since. He's published over 150 peer-reviewed publications. He's written over 40 book chapters, or been the co-author of 40 book chapters. He has served on numerous committees. He's held the highest offices, to include the highest office in our society, the Society for Vascular Surgery. He was the president in 2016 and 2017. He said that that was the hardest year of his career because he continued. Most people, when they hold an office like that, they will cut back on their clinical duties. He didn't. He continued to be the #1 RVU generator in his division and to continue to be a clinical powerhouse. He's a great guy. On a personal note, he's a fly fisherman. He owns a piece of property in Wyoming that he goes to for solace. He's on the board of the Philadelphia Zoo, which was a recent appointment. He's very keen to talk to you about that. He's an all-around nice guy. You'll never meet a finer gentleman. And another interesting note is that his son, Alex, is currently a resident in the University of Pennsylvania Integrated Vascular Surgery Residency. And he gets the opportunity to operate with his own son every now and then. So without further ado, Dr. Fairman, thank you for addressing us this morning.
Thank you. Good morning. So I'm going to take you on a bit of a journey over the next 5 hours. And it's a 20-year experience. For those of you that don't know much about the University of Pennsylvania, I joke that I'm a rather pathetic individual. I was born at Penn, and I'm still there, which is true. University of Pennsylvania was founded by, do you know who was founded? Benjamin Franklin. And this is Hamilton Walk. This is the old medical school, still there. And this is the new outpatient facility, to the left of which is going to be there's a hole in the ground, and it's going to be a new hospital there. So that gives you a little bit of the contrasts. I have no disclosures relative to this presentation. Well, I pulled some slides from history, and when I made this slide quite a while ago, but back in 1994, when I went back to Penn, I decided to put some goals down on paper. These were sort of ambitions. Maybe in some ways they were naive, but when I think about where I was in my life in 1994, I had a very huge clinical practice. I was chair of surgery at a large community hospital. I was getting, frankly, rather bored with my career. And so I knew that EVAR was around the corner, and I felt that I had sort of maxed out on what I could accomplish where I was. And so I decided, I took Clyde Barker up on an offer and went back to Penn full time. And my goal was, at that time, I wanted to learn everything I could about EVAR. I wanted to participate as a PI in every clinical trial that was coming down the pike. I wanted to obviously advance into vascular aortic therapy as it was developing. I really wanted to define my aortic practice, which I felt had become sort of boring. I wanted to make our specialty more interesting. I wanted to raise the standing of our division at Penn, which I don't think was a nationally known division. And sort of a lofty goal was, you know, I wanted our fellows to go out and become division chiefs and chairs of surgery. And I think in some ways I accomplished that. This is a slide I made up, and it shows perhaps some of the oddities about clinical trials. But so this slide, this was a patient I operated on in 1997. He was 6 weeks status post in EVAR in a clinical trial, and he was plugged into a high-risk arm of a clinical trial, meaning he was so sick that in theory we couldn't do him open. And when he left the hospital, he said to me, "Doc, what are my limitations?" And at that time, I hadn't really thought about what his limitations might be after an EVAR. And I said, "Well, I don't think you have any limitations." And here he is 6 weeks later jumping out of an airplane. High risk? Really? All right. So what do we know about clinical trials and commercial experience with EVAR? Well, we all know these things. The immediate EVAR outcomes are at least comparable, if not superior, to those obtained with open surgery. There's no ICU, there are no blood transfusions. Ben and his colleagues have popularized successfully the percutaneous option. The length of stay is oftentimes between 1 and 2 days. You can expand the therapy to high-risk patients, although sometimes if you're not careful, you pay a price for that. And in fact, those patients don't end up living any longer than they would have if they hadn't had an EVAR. The recovery is almost negligible, and you certainly can reduce inpatient costs related to a shorter length of stay. But again, you have to be careful. If you're paying more for the devices, you're going to eliminate that cost savings. What about the realities of commercial EVAR? Well, this is a huge marketplace. The penetration of EVAR in the United States is probably greater than 75%, whereas in Western Europe, where they're more conservative, probably only about 50% of patients are eligible for an EVAR based on their IFU. In the United States, the reality is we tend not to follow the IFU as carefully as we should. There will be gains going forward, and the gains are in countries, as you'd expect. China and India are going to become a huge marketplace for EVAR. The current endograft market is more than $500,000, and our own U.S. share of that is probably growing at almost 10% per year. So it's a huge market that we have a role in. One of the questions that often comes up is, does it work? Is it durable? Is it a cure? Well, sometimes it's a cure, and this is a tube graft device. We're going to talk about this in a minute. But this is an unsupported graft that never made it to commercialization. Why? Because we learned very quickly that you don't want to attach an endograft to the aortic bifurcation. That's where all the disease is. And so the attachment isn't secure. But yet it does sometimes work, and this is a man who last year when I made this slide up was 86 years old, and he had had an EVAR 18 years ago, and in fact it was still working. So it sometimes works. But then there are some realities that we need to talk about, and some of this data has been published by Andy Schanzer. We know that the outcomes long-term are better if physicians actually follow the IFU, the instructions for use. There are annoying endoleaks in upwards of 1/3 of the patients. Most of these are type 2 endoleaks, as illustrated here, through retrograde flow through iliolumbars. There's annoying sac enlargement that can occur in up to 21% of the endoleak patients. And if you look at it really carefully and critically, at 5 years, you may have sac enlargement in as many as 41% of the patients. And this typically occurs from physicians not following the IFU. They'll have an industry rep who will say to them, I'll get you through the case. A new way of sort of practicing medicine. There is an incidence of device failure, as illustrated here, necessitating revision. There's the need for lifelong surveillance. The overall cost, if you look at it carefully and critically, are in fact greater than open surgical repair. And there is a slightly, perhaps higher late mortality associated with EVAR, as illustrated here. And in fact, late rupture does remain a concern. So these are some realities that I put down on a slide about 10 years ago. And I would question whether they're really 20-year-old realities, because I think they are still realities. And that is that we can't advance technology without partnering with industry. Obviously, we have different missions than industry. The FDA approval process, and Ben knows this well, is arduous. And as I've already mentioned, off-label use is very common in the United States. We live in a litigious malpractice environment. I personally live in a city and state which is probably the malpractice capital of the world, Philadelphia and Pennsylvania. The FDA is very cautious about Class 3 device approval, and increasingly and appropriately, there are regulatory statutes, both federal and local, to protect human subject research, increasing the oversight and increasing the rules. And we all know about public disclosures and investigative reporting and new ways to access information. This has totally changed the research culture, that is clinical research, over the last 20 years. So how do we protect the public and advance the development of new endovascular aortic therapies? That's sort of the question. And compared to 1996, when I was flooded with patients at Penn who were being managed by watchful expectation of their aortic aneurysmal disease. They had been deemed too high risk for open surgery, and we all had tons of patients in our outpatient clinics that we didn't know what to do with, and it was so easy to enter those patients into clinical trials because they had no other options. And then if you now jump ahead to today, where we have 5 or 6 commercial devices on our shelves, how do we, reliably and responsibly enter patients into clinical trials for the same type of devices moving forward? I'm going to give you a little history here, and I think you'll appreciate this, but so back in the early 90s, when I was still doing a lot of open aneurysms, there was still a fear of blood transfusion therapy, and I tried everything. I had my patients auto-donate their blood beginning about a month before surgery. I had family members donate blood on behalf of the patients, but invariably the patients would end up, when they auto-donated their blood, they'd come in anemic, and the first thing we'd start doing once they were induced was to start giving them their blood back before I even began the surgery. So I was looking for other options. And I was aware of some blood substitutes, and polyheme was actually one of those blood substitutes. And this article appeared in the Wall Street Journal. One of the ethicists that was mentioned in this article was a critical care specialist at Penn, and there was concern raised about Northfield Laboratories in Chicago or Evanston that had partnered with the FDA and came up with a trial using a product, polyheme, that they actually decided to use in trauma patients without consenting them. And it was the first time anything had ever been done like this before. They actually got the FDA to approve a clinical trial whereby they would use a blood substitute in a randomized way with trauma patients based on the idea that trauma patients were so bad off, there wasn't enough time to consent them, so they were going to just do it. And they moved ahead with this trial. This was published by Tom Burton. Tom Burton, at the time, was a Wall Street Journal reporter. He subsequently won numerous awards. And this article was about we all know who Charles Grassley was. Charles Grassley, you know, the Senate Finance Committee chairman, heard about this, and he was livid. You know, what's going on here? They're not consenting patients. They're using a blood substitute. Who are these patients? Aren't these patients often indigent patients? What about their rights? And one day I'm in my office. I got a call from Charles Grassley's office. Ron, Fairman, we'd like to talk to you about a study that you did some years ago that you published. And if you read what's down below here, Northfield previously tested polyheme in a study of aneurysm surgery patients. In that trial, 10 of 81 patients suffered heart attacks, some died. None of the 71 patients on standard therapy, including donor blood, had heart attacks. Northfield halted the trial in late 2001 without completing or publishing the results. Well, what was my role in that? Well, one of the things that I did when I got back to Penn was I contacted Northfield Labs. I had read an article in one of the journals about polyheme, and I contacted Northfield Labs, and I said, I'd like to do a randomized trial in my aneurysm patients, polyheme and hemodilution versus standard therapy. And I sent them a whole protocol I wrote, a prospective randomized trial. I think we had 31 sites. They would pick the sites. And the CEO, who was a physician, got back to me and said, we're not really interested in that. And then 6 months later, he called me and said, we're doing the trial. I was very new at clinical trials at this point. I didn't know the right questions to ask. I was excited that we were going to do it. And we started the trial, and I didn't know who, it was a weird trial. I didn't know who all the other sites were. We didn't have investigative meetings. I knew that from friends of mine at Hopkins that they were in the trial. I knew from friends at UNC that they were in the trial. And things went along pretty well at Penn. And I think largely the reason they went along so well at Penn, Hopkins, and UNC was that we had really good anesthesiologists, and they were the ones that were given the polyheme and making sure the patients were safe. And then all of a sudden I got a call from Northfield Lab one day. The CEO said, we're stopping the trial. And I said, well, how come? And he said, the results are not so good. And I said, I'm not sure I understand that. Our results are really good, and I've talked to colleagues at Hopkins. Their results are good. We're stopping the trial, end of story. And I said, well, we got to publish the results. And he said, well, read your contract. It's not in the contract that we have to publish the results. So I spoke to our IRB at Penn, and actually I was allowed to publish my results. And the people at Hopkins and UNC were equally enthusiastic to publish their results. So we all wrote an abstract together and we published it. And in doing his research, Tom Burton, he uncovered that abstract. And he called me one day and said, fill me in about Northfield. And at that point I said, the only thing I can fill you in on is that we did a trial. I was upset that that they refused to publish the results of the trial, but we published our own results. And that's all I can tell you. So if you flash ahead now to 2009, this is what happened. Following FDA rejection of its blood substitute polyheme, Northfield goes belly up. They went bankrupt. They spent 20 years developing a blood substitute, including blah, 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 and the risks outweigh the benefits, and the company, went defunct. So obviously there are lessons to be learned, and what lessons did I learn? You always should publish your clinical results. Come on, whether they're good or bad, they have to be published. Issues relating to ownership of research data and disclosure of outcomes need to be sorted out before you start the clinical trial. I was never going to let that happen to me again. And you need to recognize, again, the competing interest of who's running the study and who we are as physicians. And you can't suppress bad outcomes if you're running a clinical trial. This was a huge lesson for me to learn. We live in a world today, and this is a slide, again, I made a number of years ago, but all these things are not only true today, but they're more true today. Everybody knows what we're doing. Everybody has the right to know what we're doing. Who's paying me? What What are my interests? Every talk I give has a disclosure slide, of course, at the beginning. And when we first began doing clinical trials at Penn, when I first came back to Penn, it was a different world at Penn. Penn wasn't used to doing clinical trials when I came back and expressed an enthusiasm for clinical trials. Penn was a basic science place. They had no previous interest in doing clinical trials. So the first few years I did EVAR trials, we lost millions of dollars. We couldn't bill for the devices. Devices were costing us a ton of money. But Penn actually, got their act together, and they came up with billing initiatives that allowed us ultimately for, to get billing and reimbursement for some of the things we were doing. But if you look at the history of recent technology, the same is true for TAVR. The first few years we started doing TAVR at Penn, we lost millions of dollars. And you can see that with every new technology that comes along in terms of how institutions need to catch up in order so they're making an investment, basically. Penn made an investment in me and our EVAR program, our TVAR program, our carotid stent program, TAVR, and each institution, makes those investments or decisions. About 10 years ago, I was at a meeting with Dorothy Abel, and she made this comment that I just hung on to. With everything we have learned over the last 15 years referring to EVAR, every new aortic endograft presents a new failure mode that we have not seen previously. And I thought, okay. So we'll keep moving ahead here. If this slide gives you a headache, it's actually designed to give you a headache. It's some of the things that we argued about 15 or 20 years ago. Should endografts be modular? Should they be unibody? Should they have all these different features related to their stents? Do these things actually matter? Some stents were balloon deployed. More often than not, they're now self-expanding. What about the fabric? Do any of these things actually matter? Industry would have wanted us to believe that they matter a lot. Bifurcated grafts, aortic uniiliac grafts. What about how you attach the grafts? Should you have suprarenal fixation, infrarenal fixation? What about the nitinol? What about the stents themselves? There's thermally sensitive nitinol, there's non-thermal nitinol. So all these different devices were in the pipeline that had many different features, and I would argue that probably these features didn't really matter a whole lot, but we spent a lot of time thinking about it and arguing about it. If you think about a surgical graft that you sew into the aorta when you're doing an open aneurysm, you cut the trunk of the fabric real short, you have a short trunk, long legs. In endografting, it doesn't seem to matter. There are devices of all different trunk lengths, and it doesn't seem to impact on the outcome. So many of the things that we spent a lot of time thinking about and arguing about in terms of endografting have proven not to be really all that important. This is a timeline. This is a timeline of commercialization. So these are all of the FDA-approved endografts. The ones in red are no longer commercially available. We'll spend a little bit of time talking about the Talent device. We'll talk a couple minutes about the Trivascular Ovation device here. Lombard is the most recently approved stent graft, but is not commercialized. So the company got FDA approval. They moved back to Europe. They decided not to commercialize it in the United States. But the ones in white are the ones that are commercially available today. Every one of these devices has a story. Every one of these devices actually has a timeline. And what's interesting is you can map out the timelines and see how physician feedback has changed the iterations of each one of these devices. The Trivascular Ovation, which I alluded to before, which was FDA approved in two steps, if you will, you can see here that there was an approval here and then a subsequent approval here. Well, this actually had never been done before by the FDA. The FDA actually approved the first iteration of this device using a humanitarian device exemption. And what was that all about? Well, the company convinced the FDA that there was a subset of patients out there that could not be treated with any available commercial endograft. And these were patients that had very small diameter aortic necks and small access vessels. So the FDA actually approved only that one particular device size for patients. Ultimately, they approved the whole device in 2013. But if you look carefully at what they were approving, there were only, in fact, 4 patients in the trial that had this small diameter neck. Well, what does that mean? I mean, why would the FDA do that based on 4 patients? And to me, it shows that when industry works closely and well with the FDA develops a trusting relationship with that industry partner, and they're willing to do things that they might not be able to do or willing to do with a larger company where there's a lot of turnover of regulatory people and there's not that sort of bond of trust, if you will. So I wrote this paper many years ago, and this is a little bit about failure modes, and try to keep on time here. But this was the EVT trial. This was what became known as the ANCURE device. I showed you pictures of it earlier. This was the first FDA-approved endograft. It was designed to look like a surgical graft, meaning it was totally unsupported, it was unstented. There were hooks on the top and the bottom, and the fabric in between was again unsupported. And what we learned from this trial, and which was ultimately a death knell for this type of design, is that unsupported stent graft limbs are vulnerable. They're vulnerable to twisting, kinking, and they're vulnerable in some of the types of anatomy that we treat with aneurysms. And here's an example, if you will, of a device that was deployed correctly, but if you didn't pull the device down when you were deploying it and separate these markers on the device, you would end up with a limb occlusion. Here's another example of a device that was twisted. It was unsupported and it was actually twisted. It was brought into the iliac. That would result in potentially a limb occlusion. Here's a patient, a typical anatomy, a 90-degree bend of the left common iliac artery. This patient came back some weeks later with a limb occlusion. So it turned out in this study there were primary additional interventions being done for perceived problems in almost 30% of the cases. That was a phase 2 study. And then there were interventions that were done later for patients that came back, amounting to 12%. So a large number of interventions being done for limb problems. And here's the summary. So 82% of primary postoperative limb interventions occurred within the first 6 months of this device implantation. Once you fixed it, you fixed it. They didn't come back with more problems. The problem was not fortunately associated with an outcome of amputation, but this really was the death knell for unsupported stent graft limbs. From here on in, every company came up with supported stented limbs. And you could fix these issues with routine endovascular approaches. As we got better with endovascular therapy, we routinely would be able to address these issues. There were predictors, so women, and if you oversize the limbs too much in relation to the diameter of the iliac artery, this would result in poor outcome or reduced limb flow. There was a case volume learning curve, so the more cases you did, the more likely you were to be aggressive about supporting these limbs going forward. And again, as I mentioned, this was sort of the death knell for this technology. What happened from here? Well, many of you are familiar with this. So when Guidant and EVT went to the FDA for approval, they just didn't disclose all this stuff. And so this resulted later in a $90 million criminal lawsuit against Guidant. It essentially bankrupted the company. And if you Googled at that time Guidant, you'd find multiple pages like this, lists of lawyers who were more than happy to represent you in litigation against the company. We're going to talk a little bit about the Talent stent graft, and I mentioned to you that every device has a story. I don't know if, Ben, you know what Talent stands for, but Talent was a partnership between 2 people. There was a general surgeon in Buffalo named Tahiri who came up with the idea of the Talent stent graft, and he partnered with a guy named Howard Leonhardt. Leonhardt was an entrepreneur with a community college education, a very strong people person, and the 2 of them started World Medical Company, Talent, Tahiri, Leonhardt. And they were a worldwide company. They had plants all over the world. Back in the mid-90s, they were producing talent stent grafts. This sort of got our division at Penn on the map. We did a huge number of these cases. There were failure modes with this. Here's an example of an acute seam separation from over-ballooning using a compliant balloon. Here's an example there. There were stent fractures, and I would argue that almost every endograft has developed stent fractures along the way. This device had a connecting bar, and if you didn't orient the connecting bar well, you would result in a bend or a kink and an occlusion of the limb. And you needed this connecting bar at the time. It provided columnar support for the device. So you couldn't get the device out of the delivery system without this connecting bar. But what companies do when this happens is they buckle down and they do a root cause analysis. And they figured out what the problem was. And the problem was with the nitinol. The nitinol had imperfections, and when they refined the process of manufacturing these nitinol stents, including a process called chemical etching, they were able to eliminate the future fatigue fractures that these stents were developing. Well, this, as I mentioned, this sort of put us on the map in the late 90s, early 2000, and yet the device, as I mentioned earlier, wasn't approved to 2008. Well, what was the deal with that? Well, in those days, if you had a patient that needed an EVAR, you could call Howard Leonhardt at World Medical and say, I've got a really sick patient. I need one of your devices right away. He would get a device sent to your office. He would send an engineer from World Medical, and they would take you through the case. And so there were a huge number of these cases across the country that were being done without any follow-up. And the company then changed hands. Leonhardt sold it to AVE. He bought a winery in California. He later ran for governor of California. But then it turned over again to Medtronic. And so when Medtronic, this was a really good device. And when Medtronic went to the FDA to get this approved, the FDA said, show us the data. Well, there was no data because they had no follow-up for these patients. So eventually, with the help of Mike Marin, who had his own IDE, as well as a new study, the device ultimately went forward and was FDA approved. What did I learn from this? Well, I learned many things from this trial, and as I mentioned, this sort of put us on the map at Penn. We had done, I think, over 150 implants, and when a device goes to the FDA for approval, they look at the sites that did the most patients, and you get audited. And so one day, an investigator from the FDA, showed up in my office and seemed like my best buddy and said, Ron Fairman, we're going to audit you so that this device can be approved. They spent 3 weeks there nonstop. They went through every piece of literature, every documentation that I had, went through every patient's chart. And in the end, we were cited because there was one patient that they felt I should have entered in the high-risk arm, and I entered the patient in a low-risk arm. But if you're going to do the most EVARs in a clinical trial, you need to be prepared to be audited. It's not the worst thing, but that's what happens. Okay. Some of you are aware of the Gore excluder device, which is widely used today. This is an example of fabric permeability failure. When the device was first used in the clinical trials, if you looked at what was happening over 5 years, you can see the sacs are growing. That's not supposed to happen. And they did a root cause analysis. They found that it was the permeability of the Gore-Tex material. They went back and refined it, made it a low permeable design. And you can see the problem of sac enlargement went away. The test group is the low permeability version. So further refinement of devices over time when you deal with companies that are very responsible. The Anaconda device, I was the PI for this trial with Jeff Carpenter, one of my former partners. Jeff is chair of surgery at Cooper in New Jersey. So the Anaconda stent graft system was already CE marked approved in Europe when they came to the United States and wanted to do a first-in-man study in the United States. We jumped at the experience, and we did a few cases, Jeff and I, And we immediately began to have problems getting the delivery system out of the patient. So we would deploy the device, and the delivery system would be stuck in the patient. It wouldn't release from the device once you deployed it. And we essentially would break the device to get it out of the patient. We did about 2 or 3 cases. This seemed to be a recurring problem. We mentioned this problem to the leadership of Vascutech, who was running the trial, and said, "Oh yeah, we know about this. We had this in Europe, but we don't have that device iteration anymore. We now are on device iteration 3 in Europe, but when we came to the United States, we didn't have enough inventory, so we brought device iteration delivery system 1 to the United States for this trial." Well, that was the end of our participation in that trial. There was no way we were going to put another one of those devices in. And they later came back to the United States with a phase 2 trial that they ultimately did. We chose not to participate in it. It was never commercialized in the United States, but the device is still being used in Europe. And that's the lesson. That's the obvious lesson. Ask the right questions, find out, you know, if it's already in use in Europe, what have they learned in Europe, and what are they bringing to the United States? You all know who this is, Juan Perotti. This is a time when I made this slide that my sort of confidence and arrogance sort of got away with me. So obviously this didn't happen in 1999. I thought it was going to happen, but it didn't happen. And this was a new device. It was a cordless device, 12 French sheaths, first in man. This is a pre-op. That's the post-op. Boy, that just looks perfect. Trouble is, a couple days, the patients came back with limb failure and endo leak. This is the design of the device. It's a weird design, a polyurethane gasket with very flimsy limbs that were stuck in it. In animals, polyurethane rapidly became incorporated into the aorta. So the idea was that when you put this in a patient, within a month it would be incorporated. There'd be ingrowth. It would be very novel. This was a total failure of concept. And at the time, you know, my eyes weren't opened wide enough to really follow my intuition. I had to explant all of the cases we did at Penn. Thankfully, nobody died. There were 2 ruptures. But you can see what the problem is here. Here's the polyurethane, big holes where there was no seal. And if you tried to balloon these limbs to seal the endo leak, the holes became actually larger, a failed concept. We're going to talk a minute about trivascular, and I hope I'm doing okay here. This is not the trivascular ovation. This is the predecessor to that, the trivascular graft. It was a company that was started by a few very smart engineers. This was the design, a very novel design. It used a biopolymer to create seal, an incredibly robust suprarenal nitinol skeleton to hold it in place. I had never seen anything this robust before. But I thought it was unique, and this is what it looked like at the time when you would fill the biopolymer. You could see it radiographically. It would cure very quickly, and over about a month or so, this would disappear from your radiographs. The phase 1 results were awesome. I mean, we thought this was the new future. And what happened is the engineers sold the company to Boston Scientific. Boston Scientific decided they were going to plan a phase 2 trial. They were getting underway to do that when all of a sudden, all these stent fractures started popping up in that suprarenal segment. And the reality was, it was such a big, stiff suprarenal segment. When you put that in the visceral segment, it was a setup for fracture, for stent graft fracture. The FDA said to Boston Scientific, you bought the company, you figure out a way of following these patients. Boston Scientific, without any feedback from the investigators, said, we want you to follow the patients every 3 months with a CAT scan indefinitely. Think about the practicality of that. There is no way my patients were going to come back to me every 3 months for a CAT scan. And I knew that if they didn't come back to me and they were still in the trial, I was going to be set up for protocol violations. Well, you might think, well, what's the big deal of a protocol violation? It is a big deal. And so I asked the IRB at Penn, what do I do? Should I let the patients drop out of the trial, which is what the patients wanted to do, and then I could walk away from it, or accept the fact that they weren't going to come back, try to get them back every 6 months, every 9 months, and accept the protocol violations. The IRB at Penn said, take the protocol violations. So that's what I did. These are what the fractures look like. It's just of interest. You can see it radiographically. They're easy to see. And what was the outcome? Well, when these fractures occurred, that was what was holding the device in the aorta. So these devices went south when these fractures occurred. This was a big deal. This was an international study. At Penn, I had enrolled more patients than anybody in the world. It was only 11 patients. One day, I was coming back from the OR, and I got to my office, and there was a registered letter on my desk. The letter was made out to the Penn IRB from Boston Scientific with a copy to the FDA, and it said to the head of our IRB, dear so-and-so, we want to bring to your attention that one of your investigators, Dr. Ron Fairman, is not in compliance. He has more protocol violations than anybody in the world in this clinical trial, only 11 patients, and we would suggest that he not be allowed to do clinical research anymore at the university. Copy Dorothy Abel at the FDA. And you can imagine what my reaction was. I called the hospital attorneys. I had documentation from our IRB that they had encouraged me to keep the patients in the trial, take the protocol violations, and that the university wrote a letter to Boston Scientific, sort of a cease and desist letter, and my life kind of went on. But again, a lesson to be learned, as illustrated here. A few more little Ron Fairman experiences, and we'll wrap it up. But the Aptus modular endograft was a stent graft that was not much different than other stent grafts. It had supported limbs. It was an unsupported midsection. What was novel about this was that it used screws, endostaples. You'd screw the graft into the aorta with the idea that it would never migrate. Very, very novel idea. Low profile delivery system. And this is what the little screw looked like, about this compared to the size of a dime. I presented this at the VEITH meeting in October of '09, and what happened with this device, the sad thing about this device was the most novel feature of the technology, the screw, worked okay. But the stent graft itself was a debacle. And in 2009, I presented this data where about 30% of the patients in this trial were developing very serious thromboembolic events. These were not claudication events, these were amputation events. And they were developing these events at weird times. They could be running on a treadmill or jogging in the neighborhood, and they'd all of a sudden develop a limb occlusion and fall down. And what was happening when we, as illustrated here, they did a root cause analysis, they hired Jeff Elkin, who was a former engineer from World Medical, very sharp guy, and he found out that there were in fact manufacturing inconsistencies at the company resulting in high shear stress, platelet aggregation, and embolization. So when they were running, they were putting themselves in a high shear stress situation, and an endograft junction where the trunk attached to the legs of the endograft, where it was narrowed because they were not following the manufacturing specifications and they were producing them too small, they would develop an acute limb occlusion and have a thromboembolic event. Huge problem. The sad part is there was nothing attributable to the endostaple technology at all. After I gave this talk, my friend, becoming my friend, Tom Burton, who was in the audience at the VEITH meeting, the Wall Street Journal reporter, came up to me and said, tell me about the Aptus device. And I said, I can't talk to you about the Aptus device beyond what I said. I have a confidentiality agreement. Call the company. So Tom Burton called Jeff Elkins. Jeff Elkins regaled him about the technology of endostaple. And then at the end of the conversation, Tom Burton said, tell me about the limb failures. And then Tom Burton wrote this article. And it's absolutely true what he wrote, that the FDA, and I didn't know this, but the FDA doesn't go into companies during the conduct of a clinical trial and inspect how they're manufacturing grafts. They don't do that. They may do it post-market, but they don't do it during the conduct of the clinical trial. So what is my responsibility as a PI? It turned out the person that was running this trial and the person that was running this company had no prior aortic endograft experience. This was not a Medtronic or a Gore. This was a startup company where the CEO had done some peripheral trials before he became CEO and running this trial. So you do have a responsibility. I felt a responsibility to know what's the experience of the people that are actually running this trial. And that's impacted on me ever since then. This is a sobering slide. I made it up and I've updated it periodically. But these are more recent clinical trials. And what's interesting that is we're still seeing limb occlusions, even recently. This device was recently finished clinical trials. I would say that this will never be FDA approved. Very questionable whether an Endowed device will be FDA approved. If it ultimately is approved, it'll be in a very limited subset of patients with specific anatomy. I ran 2 trials as PI for a Zenith low profile device. Both trials failed miserably, as illustrated here. We stopped the trials. So these failure modes continue with new device iterations, just as Dorothy Abel predicted they would. We presented this at the SVS last year, and I'm going to wrap up with this. But this is some work that my son Alex did. But he looked at EVAR devices that we did at Penn over a 16-year period. These were not EVARs that were done anywhere else. These were EVARs that we did, and we followed the patients. It was not our fenestrated or branch population. And you can see that we divided reinterventions into those with FDA-approved devices, those with trial devices. Our overall reintervention rate was about 7.5%. And if you look over the 16-year period, the trial devices had an increased incidence of reinterventions here, as illustrated here, to 6% or so with FDA-approved devices. But what perhaps is most sobering is if you look at trial devices developed into divided into 2 8-year cohorts, we're not getting better. So trial devices in the most recent cohort, when you would think we've learned so much from prior years, are actually worse in terms of reinterventions. This looks at the number of reinterventions for trial devices, again, slightly higher versus FDA-approved devices. What's perhaps sobering is if you go out further in a timeline, you will see that even 8 years after implant, patients are still undergoing reinterventions. Most of the reinterventions are as you'd expect for type 2 endoleak in both FDA and trial devices for growing sacs. With the FDA-approved devices, we're more likely to have type 1 endoleaks because maybe we don't follow the IFU as well as we should as a clinical trial patient, which is more vigorous. And again, I've alluded to this earlier with the increased incidence of iliac limb occlusion in trial devices versus FDA-approved devices. It's an ongoing clinical problem. These are some of the procedures. In most cases, we're doing translumbar embolization, iliac angioplasty and stenting, and proximal and distal extensions for the reinterventions. The number of reinterventions was definitely associated with the need for explanting these devices, but in fact, the type of device, the etiology of the reintervention, and the type of reinterventions were not predictive of the need for explant. And mortality did not actually differ between study cohort or the need to explant. So to wrap it up, where are we today? I do believe that we as PIs, and if you followed me on this journey, hopefully over the last 20 years, I've learned some things as well as increased humility as I've gotten older. But we do have greater PI responsibilities, I think, than we did 20 years ago. We don't have tons of patients in the wings waiting to die of ruptured aneurysms. We have some, but when you enter patients into clinical trials today, you really have to sort out some issues. You know, what's in it for the patient? Is this new potential design something that's going to actually benefit patients as opposed to benefit my career by advancing me academically? If the device is already in use in other parts of the world, what version of the device are they bringing to the United States? And finally, as Dorothy Abel said in a haunting way years ago, new devices do bring new failure modes. So I want to thank you all very much. You have been wonderful hosts over the last 2 days. I will definitely carry these memories with me, and thanks for staying awake and your attention.
All right, questions for Dr. Fairman? Yes, ma'am.
Comment, I enjoyed your talk, and lessons learned obviously should hit home with especially the junior members of the audience as they launch their careers. One of the things that you allude to as academic surgeons, I think we're all really excited when a new product comes along and we have the opportunity and an industry comes and challenges us, and you danced around this topic of cost associated with that. And I think our enthusiasm to participate often ends up with our under-appreciating how much it costs for us to do that study. And you talked about the technology associated costs, but I think that as people in the audience do participate in these studies, they need to strongly consider the human costs as well, because the research coordinators that you need to hire to do all of that follow-up are expensive.
Yeah.
And learning to budget appropriately for these is a very important lesson, and so I appreciated your comments about that.
Yeah, when I first started doing clinical trials back in the mid-90s, there was so much low-hanging fruit, it was easy. I think when we first signed on to the Guidant EVT trial and the Talent trial, we were paid a lot of, not me personally, but our research program was paid a lot of money per patient entered over 5 years. And then if you flash ahead to today, the amount that they will pay to support a patient in a clinical trial is minuscule compared to that. I think, you know, we were getting somewhere along the lines of $30,000 a patient back then when we entered them in a clinical trial. And that was designed to cover their costs over 5 years. It's minuscule now compared to that. We have, one of my other jobs at Penn is I'm chair of the corporate supply chain for the whole health system, which has become sort of a huge job. And so one of the things that we put in place is that if we have commercial devices already on our shelves and someone wants to bring on a clinical trial of a comparable device, that we mandate that the cost of that device cannot be more than what we're already paying for commercial devices. Now, you could argue aggressively that that cost should be less than what our commercial devices are, but that's never going to fly with industry. But at least we have a mandate that we won't pay more for an investigational device if we have something similar on the shelf that's already commercially available. And that seems to work in our health system. But you're right, the costs are huge.
Other questions? Dr. Wood?
Dr. Fairman, this was great. And I'll say that as a native of Michigan, cheering for blue on Monday, the dominance shown by Nova Nation was humbling, as is the dominance of your work in the endovascular world. So I was pretty disappointed on Monday, and I know that you guys were cheering, but so there were some of us that were devastated. You've given a great kind of journey of successes and failures in partnership with industry, and that's always a discussion that we have with residents and junior faculty and in clinical trials of there being substantial advantages of being able to have partnerships with industry, but you've also demonstrated some lessons and some pitfalls.
Some dark sides.
Yeah, and do you have any advice for our junior faculty that are kind of relatively new or novices in partnering with industry, of navigating that minefield of maybe very experienced industry partners, that, and how to avoid being taken advantage of in this aspect of clinical trials with industry?
Yeah, well, I hope that, you know, based on some of the lessons that I've learned, that people will think before they leap. You know, it's hard. I mean, you're in a competitive academic environment. You know, you as a young faculty member want to succeed. You know, there's an immediate tendency to be overjoyed that an industry partner, especially a big industry partner like a Medtronic or some of the others, would want to work with you and do a clinical trial. There are definitely competing interests. You can easily see how this, you know, elevated my academic career by doing this. It led to me give presentations at VEITH, me getting known around the world by doing this stuff. But you have to sort of be grounded. I mean, you have to, you know, stop, think about what you're doing, and oftentimes your immediate impression is correct. That first Cordis device trial, when I looked at that design, I thought it was a piece of crap. You know, that polyurethane gasket, it was flimsy. I thought, how the hell is this going to work? I should have stuck with my gut feeling. Thankfully, you know, nobody died, but it was very sobering. And the thing that I found over my career is I have less and less tolerance for complications. That may just be true of all surgeons, but you know, I had a bigger stomach back then. And I thought that I was justified for feeling that way because we had lots of patients that had no alternatives. And so I felt there was this higher imperative. You know, why not put them in a clinical trial? They're going to die anyway. Let's put them in the clinical trial. And many of them did great. You saw that slide of that 18-year-old endograft. And so, you know, it was exhilarating. But on the other hand, as you get older, you have less of a tolerance. When I did the Aptus trial, and I found out when they did the root cause analysis that there were manufacturing inconsistencies, I was furious. You know, I wanted to talk to Tom Burton, but I wasn't allowed to, but I was furious. And that's kind of part of the maturation process. Everybody who does this is going to go through that. You're going to reach a point where, you know, you don't want to see patients get hurt. And when we look at our own results at Penn, and we now see that newer iterations of investigational devices are resulting in increased reinterventions compared to our commercial devices on the shelf, you know, where do we go from here? So, yeah, I just think, you know, young faculty members, young trainees, you'll all be faced with this. You just have to get your arms around it and figure out what you're comfortable with.
I'll finish up with just, do we have one more question? Wayne?
Dr. Perelman, it's great talk. I learned a lot from your talk. The question is, how reliable industry sponsored trials, and how FDA approved the new devices? Because we have so many devices approved by FDA, but after a while, you know, clinical practice, the proof has failed, and they have to go a lot of money.
Yeah. I think there's a new paradigm for that, and that is with the SVSVQI, which is our National Society Registry. So more and more, when the FDA approves a device today, there's going to be a post-market registry. And so that'll give them real-world experience, which I think is what you're referring to. So, you know, running a clinical trial and getting good results is one thing, but then how that device behaves in clinical practice is totally different. And increasingly, the FDA, the TCAR project is an example of that. So when the FDA approved TCAR, which is a new carotid stent system, they partnered with the SVSVQI, so there's a post-market registry that will provide them with real-world data. And I think that's the answer to that. You're going to see more and more of that with device approval, where the devices will be continued, they'll be continued to be tracked over time, leading to more information in terms of outcomes.
I'll finish with one final comment and a question. I, like many others, held the opinion that the FDA was this obstructionist agency for getting devices approved, and really did not have a high opinion for the FDA up until the time that I applied for my own investigational device exemption clinical trial. And what I found is that the closer you work with the FDA, the more open they are about different things, and they're actually very easy to work with. So my opinion of the FDA has completely changed, and I think the FDA is getting better at the approval process. They have this new designation of breakthrough technology, and I wonder if you might comment on your experience over the last 20 years of working with the FDA in 1996 versus working with them today.
Yeah.
Has it improved, or?
Well, I think you're absolutely correct about that. What I've seen with the FDA is they like to form relationships with people. And so when Medtronic had a period of time where every other year their regulatory people were coming and going, the FDA could not embrace that at all. They didn't feel they had a trusted partner to work with. The FDA bitterly resented what happened with the Anurex stent graft, where they felt that Medtronic was disingenuous when that whole trial was done. It led to FDA approval. So when the FDA can form relationships with people like you or industry, for the last 10 years, Medtronic has had a very stable regulatory group. They trust each other. They work closely together. Vascular was a great example of that, because there were 3 engineers who could work very well with Dorothy Abel, and that's what led to that unusual humanitarian device exemption approval that had never been done before. So I do think, you know, and Dorothy Abel's a wonderfully smart, engaging individual, but it's not a pushover. Yeah. And, you know, I agree. I have great admiration for what the FDA does today.
So we'll finish with presenting Dr. Fairman with a token of our appreciation. This is a textbook that we authored. There's not another one like it in the literature. It's solely devoted to ruptured aortic aneurysms. And the unique feature of this is that it has, one of the authors is your very own Ben Jackson, who partnered with one of your mentees, Derek Nathan, who was one of our vascular fellows, to author a chapter. And I want to present you with this.
Thank you very much, Ben.
Thanks, Ron. All right, thank you, everyone.